So, in the previous class, I had given an introduction to what classification is. Just a classifier, I just opened a note and just for you to revise certain basic concepts, and also to get us introduced among ourselves of how we will be teaching and all these aspects. So today, actually, I'm going to start my classes in a serious manner, or not a serious. I would. The better word is in a systematic manner. So today's class basically it starts with the basics of classification. Before we start, I would like to remind you people that always try to consult your NCERT class 11 and 12 textbooks. If you have NCERT, then you don't need to buy, otherwise buy these NCERT textbooks, because that textbook, NCERT chemistry and NCERT biology, those textbooks are very essential for keeping your basic concepts. These concepts are going to be extended. You will read more about it, what you have read in your class 11 and 12. Ecology is one chapter this year which you will be studying, and this semester, and principles of classification and non-coordinates. All these things you will find in this NCERT textbook. Other West Bengal textbooks, they are also fine, but if you don't have, if you are from a Bengali board, I would recommend you buy the English NCERT textbooks 11 and 12. That will help you to get into the basics very carefully in English. Although you have studied it in Bengali, if you have studied it in Bengali or Hindi or from whichever medium you have studied, once you study this, you will find that this is absolutely the same thing, and you will adjust very slowly to English. Let me tell you, you have to improve your English, both spoken as well as written as well as understanding. Right now, whatever, if you are from a Bengali medium, whatever you are reading in English, in your brain you convert that into Bengali, and then you try to understand this thing. But that is something which you should stop. You should start thinking in English. This is the most important thing, because you have to write in English in your examination. So this way you can specifically train yourself. It's nothing complicated. There are so many students year after year, they have done it. I have seen them. They have done very well also. From Bengali medium, they are doing very well. They have done well, and they are doing well also. So let us start today's class. Is my slide visible? One of you unmute and tell if it's visible. Can you see it?
Yes, sir.
Okay.
Yes, sir, visible.
I can see it.
Okay, everybody can see it then, fine. And also let me remind you that for example, if you are having a problems with English, you ask the questions in Hindi, ask it in Bengali. If you have problems, just stop me and say that, sir, আমি বুঝতে পারিনি এই জায়গাটা, বা স্যার মুঝে ये जगह समझ में नहीं आया. I will of course explain. I can speak both Hindi as well as Bengali. I can explain it to you. So that should not be a problem. But one thing for sure, I can explain it in Bengali or in Hindi, but the basic class I will do it in English, because you people really need to get into English from day one. It has to be from day one. Even when college reopens, please request the teacher to speak in English. If they start speaking in Bengali, many people who don't understand Bengali properly, they will face problems. So just very politely, sir, I don't understand Bengali. It would be nice if you could explain it in English. In that way all problems will be solved. So if a teacher is unable to explain to you, the teacher himself or herself will feel bad that I am being a teacher, I am unable to teach the class. The teacher will never be speak with you badly and say that I will speak in Bengali. He or she will not do that. That person will try to explain, the best you can understand. That is the natural quality of a teacher, and that anybody who starts teaching students, this thing automatically, this type of attachment automatically comes. So you don't need to worry about it. Okay. So basics of classification part 1. This picture has been taken from the class 11 textbook, NCERT. This particular picture is there. You can see that there is something known as kingdom, animalia, multicellular. Then you have another term used, levels of organization. There is another term used as symmetry. There is another term used as cavity or coelom, and another term you find phylum. So whenever you speak of kingdom, the first thing which you see surrounding yourself is either plants or animals. So this is animal. You're talking about animals. So this is animal. Then you go to a level of organization. The level of organization can be a single cell, one cell, or it can be a tissue. A tissue is a cell which has got a specified function. Then you have an organ. Several tissues together form an organ. Several organs together form an organ system. So this is known as the levels of organization, whether it's cellular level, tissue level, organ level, or organ system level. So these are known as levels of organization. The word you use is levels of organization. When you talk about a cell, you are talking about a cellular level of organization. When you talk about, say, liver tissues, liver cells, that is a liver cell is a tissue. So you are talking at a tissue level of organization. When you are talking about the liver, the whole liver having different types of tissues, then you are talking at an organ level of organization. When you are talking about the digestive system, it's an organic system. You have your liver, pancreas, stomach, intestine, all these organs together, they are known as an organ. This is the organizational level of level. Okay. The next thing in animal system, which is very important. Bolo. Any questions?
Sir, so basically the kingdom Animalia are divided into two parts, unicellular and multicellular.
That is another level of organization. That is another type of classification. Okay. They can be unicellular. There are many ways you can define the living kingdom. You can do it as unicellular, you can do it as multicellular, you can do it as plant, you can do it as animal, you can have a 3 system of, you can have it as protists, you can have it as monera, you can have it as animalia, you can have it as planta. So there are different ways by different people. I will talk about these things. Different ways by which different people have classified the living kingdom. Right. So for your syllabus, right now the current syllabus which is present, you need to study about the 5 system classification, and rather you don't even have to study about the system of classification. But to start classification, you have to start with, for this moment, at this moment, think that it is a 2 kingdom classification, plant and animal. So plant can be unicellular, animal also can be unicellular. Plant can be multicellular, animal can be multicellular. It is like different people have classified it in a different manner. The people saw life in a different manner. Some scientists saw that life in 2 manners. Some people thought that okay, only plants and animals division is perfect. Some said that oh no, plant and animal is different. What about bacteria? Are they plant and animal? Oh no, they are not plant and animal. So okay, then for bacteria we have one kingdom. Kingdom bacteria, kingdom animalia, kingdom plant. What about fungi? Then some people say, are they plants? Oh, they are not plants. So for fungi we must keep some another kingdom. So like this, different people have given different ways, but none of them are wrong, and none of them are perfectly correct. So basically you have to study all of them. You have to know all of them, and basically you have to know why you are studying this classification, and Also you must know why you are not studying this classification. क्यों इसको पढ़ रहे हो इस क्लासिफिकेशन को, और क्यों नहीं पढ़ रहे हो, ये दोनों चीज याद रखना है। এই দুটো জিনিস তোমাদের মনে রাখতে হবে যে কেনটা পড়ছো, কেন পড়ছো না, কিন্তু but you have to study all of them. And the levels of organization I have mentioned. Then you have another thing known as symmetry. The symmetry can be radial, I will explain very soon, or the symmetry can be bilateral. Then another thing we have to study is a body cavity called coelom. I will explain. So there is a special body cavity. Animals have got a special, the animal can be divided. If you cut an animal, you can find 3 layers of cells, tissues, cells. One is known as the ectoderm, then you have the
But sir,
mesoderm. Follow?
but sir, when fungi become detected as it is, and মানে এর ক্যারেক্টারিস্টিক্সটা পুরোপুরি মানে প্ল্যান্টি বা অ্যানিমালিয়া এদের তুলনায় আলাদা, তার জন্য ফাঙ্গিকে একটা আলাদা ক্লাসিফিকেশনে দেওয়া হলো। তার নাম হলো ফাঙ্গি, প্ল্যান্টি থেকে এগুলো আলাদা হলো।
ওটা হচ্ছে দ্যাট ইজ বিকজ ইট ইজ একরকম একজনের ক্লাসিফিকেশন, আরেকজন কিন্তু ফাঙ্গিকে প্ল্যান্টের মধ্যেই ফেলেছে, ঠিক আছে।
আচ্ছা, আরেকটা দিকে মনে রাখতে, যেখানে ব্যাকটেরিয়াদের ইনক্লুড করা হচ্ছে সেখানে প্রোটোজোয়াদেরকে প্রোটিস্টাতে ইনক্লুড করা হচ্ছে। বাট স্যার তাহলে
so no, bacteria and protozoans are different. Bacteria are prokaryote organisms, they don't have cell organelles, and your protists, they are eukaryotes, they have cell
protists are eukaryotes.
Exactly.
But sir, my question is, তাহলে মানে 5 কিংডম ক্লাসিফিকেশন যেটা আরএইচ হুইটেকারের, বাট স্যার তাহলে ভাইরাসকেও কেন এর সাথে ইনক্লুড করা, মানে একটা আলাদা ক্লাসিফিকেশন ভাইরাসের কেন তৈরি করা হয়নি?
Viruses okay, the question is why viruses don't have a separate why viruses are not grouped in one of these kingdoms, maybe kingdom virus. Why don't we have a kingdom virus? Okay, the question is why don't we have a kingdom virus? It's a very good question, because viruses have got a very strange properties. When they are outside the body, they are non-living, as well as it can be a grain of sand. Why? Well, the protein which you eat, is it living or is it non-living? It is non-living. So why don't you have a separate kingdom for protein? Why don't you have a separate kingdom for carbohydrates? Why don't you have a separate kingdom for vitamins? We don't, because they are non-living. So outside the cell they are non-living, because they don't show any symptoms of life. No life at all. That whether even when you have bacterial spores, the spores, they show minor level of organization, some sort of life, some respiration, some digestion, something of life is going on. There are several things of life going on, some biochemical reactions, something about life is going on inside spores. But these viruses, nothing goes, so they are non-living basically, right? So only when they come inside the body, they show certain characteristics of life. Therefore, how do you define life then? You cannot classify. So for virus, you have a separate group of studies about virus. Although we study whether some people had raised the question whether we should study virus in biology, because basically when they are inside your body, they are non-living. Inside your body they are living, and even the living, they don't have a body also inside your body. Viruses, they don't have a body in your body. The funny thing about viruses is when a virus attacks a cell, okay, so that non-living particle, it has got a coat, cellular outer covering. Inside the covering you have either DNA or RNA and one or two types of proteins. So it enters your cell. What it does, the cell when it enters your body, what it does inside your cell, it just transfers its DNA or RNA inside the cell, nothing else, and that DNA gets integrated into the cell, okay, a cell of the host. So you have a one of your lung cell. In your lung cell, a virus comes and infects your lung cell. So the virus never enters. What enters is only the DNA. So the body of the virus never enters your cell. Again, it's non-living. So it's the DNA which enters, and it enters inside the host cell, and inside the host it integrates into the host DNA, and it all the processes which take place after integration into the host DNA, you will find that this is taking place with the host machinery. The virus have got nothing to do. So how do you can you classify them as living really properly? How do you classify them in a kingdom? You cannot show any essence of life. You can show characteristics. So for them you have a separate group. You cannot place them under bacteria. You cannot place them under thing. You cannot even place them under completely living organisms. Therefore viruses have where do you place viruses? You can't keep them separate. So viruses are studied in a there is a new system of classification where virus is placed in a different kingdom itself, which has got no relationship with your other kingdoms. Viruses, now there is also another aspect which you must study, and that is the aspect of evolution. So all these classifications, they have got an evolutionary thing. It is presumed that something, the earliest thing which to come were the archaebacteria, archaea. So these archaea evolved and they formed the bacteria, the eubacteria. These bacteria, somewhere down the lane, they became unicellular animals like protists. These protists, somewhere down the lane in evolution, they became into multicellular organisms. These multicellular organisms, they then divided into something like plants, and they divided themselves something into animals. But viruses, they perhaps are super bugs. They came much later after animals had come. So they are not very primitive. Rather, they are more advanced. Even after animals came, the viruses came. So if you study, you cannot place a virus properly in the evolutionary chain. So there are a lot of these problems because of which you cannot include virus in your classification. This is a very good question. So let me go to the next slide. Okay. This is the two kingdom classification by Linnaeus in 1758. This famous Linnaean system, Carlos Linnaeus, the famous system of classification, plant and animal. Actually, he did not give the name plantate. The name was Vegetabilia. So the name came from vegetables. After this, you come across to the levels of organization, cells, tissues. So these are different cells of the leaf. They are known as leaf tissues. Even for liver animals, you can say this is the skin tissues. Then organs. Then all the leaves together along with xylem and phloem, they form the organ system. Here you are talking about the circulatory system of the horse and the whole organism. So this is how you define the different levels of organization. Then we are talking about symmetry. This is a picture of a sponge. You cannot divide. Symmetry is how you divide the organism. Then you have a cnidaria or these corals. You can divide it here. If you divide it from this angle, this plane where my mouse is moving, you can divide it into one half here and one half here. Let's make a laser pointer over here. You have 1.1 half here, another half here. From this plane if you divide. Now if you divide from this plane, you'll have one half here, one half here. And both these halves, they look the same, they are equal. Then so from 5 planes you can divide from different, wherever you divide, you'll find two parts are equal. But in this insect you can divide it from the half. So this type of symmetry is known as radial symmetry, and this type of symmetry is known as bilateral symmetry. This is symmetry. According to this, you can divide organisms. Then the important thing is the coelom. There you have organisms can be classified as coelomate, pseudocoelomate, or acoelomate. So in a coelomate organism like earthworm, if you take a cross section, this cross section, you will find an outer layer called ectoderm. Then you have a mesoderm, and you have an outer mesoderm and an inner mesoderm, the red color. in between the mesoderm you find a fluid filled cavity. There is a lot of fluid, joliyo padarth. Then that is known as coelom. And inside you have the yellow color endoderm. So like from earthworm and higher organisms, they have coelom and they are known as coelomates. But when you talk about your roundworms, your like ascaris, ringworms, if I find or filarial worms, they are pseudocoelomate. They have a space, but that space is not in between the mesoderm. It is in between the mesoderm. It is between mesoderm and endoderm. Here you see the space in between the red layers, that is between the mesoderm layer. And here you find the space in between the yellow layer and the red layer. So this is a false cavity, therefore they are pseudocoelom. And you have your tapeworm, or this is Digenia. These are free living helminths, like tapeworm or
planaria.
Planaria. They all belong to this acoelomate class. So they don't have any space. Ectoderm, mesoderm and endoderm, they don't have any fluid filled space. So you can divide animals like that also. Then you can divide the animal kingdom into phylum, subphylum, classes. এখানটা একটু সরিয়ে দিচ্ছি এই জায়গাটা এখানে. You can broadly you can divide it. So the animal kingdom you will find annelids, the earthworm like structures, kencho. Then you have the nematodes, the roundworms. They are dangerous. When you are a kid, perhaps you had it in your stomach anytime. Then you have the arthropods, which include the insects and your chingri mach. Then you have the platyhelminthes or the flatworms, the tapeworms. Then you have the chordata. The chordates have got vertebrates. They can be divided into amphibians, reptiles, birds, mammals, bony fishes, cartilaginous fishes, lampreys and hagfish. Then they can be poriferans. They can be echinodermatans. They can be cnidarians. The echinoderm are the starfishes, the porifera are the sponges, the cnidarians are the coral reefs and the jellyfishes. They are not exactly fishes. They look, this is a jellyfish and this is a coral. Then you have the mollusks, the snails. And of course you have the among the mammals, you know, all the amphibians, the reptiles, the birds, the different fishes. So this is basically the animal kingdom. Okay, so I come back to this part, that is kingdom animalia. The হ্যাঁ বলো.
এর আগের স্লাইডটাতে যেখানে আপনি ফিশ লেখা ছিল ওটাই তো অস্টিকথিস ক্লাস.
হ্যাঁ ওগুলো পরে হবে. We will talk about those later. So right now don't worry. Yes, ostrictis, chondrichthyes, all these things that are further classification. So we come back to this again. You have a kingdom animalia, you have levels of organization, cellular or tissues. You have the symmetry, it can be radial, it can be bilateral. You have a body cavity or coelom, without body cavity, with false coelom, with true coelom. You have the poriferans, stenophorans, cnidaria, platyhelminthes, ascihelminthes, and these are the ones which with the true coelom. So the same picture I'm going again and again. So now we talk about what is biological classification. So the 5 kingdom classification of living kingdom, it was given by Whittaker in 1969. They divided it into monerans, which are unicellular prokaryotes, the protistans, unicellular eukaryotes. Eukaryotes have got cell organelles like nucleus, ribosomes, Golgi body, mitochondria, all these things are present, therefore they are eukaryotes. True karyon means cell. They are true cell, and protists, they don't have a true cell, like bacterials, monerans. Then you have fungi, plantae and animalia. They are multicellular eukaryotes. So what is biological classification? Biological classification or zoological classification, it is the process by which scientists group living organisms or animals. So it is the process by which organisms are grouped, living or non-living. Organisms are classified on the basis of how similar they are. If they are similar, they are placed in one group. If they are not similar, they are placed in a different group. So, like I did yesterday, the last class I mentioned the difference between systematics and taxonomy. Systematics or systematic biology, it is the field which first provides scientific name for organisms. You provide a scientific name. For example, humans are Homo sapiens. So, anywhere in the world, some people call insan, some people say manush, some people say Leute in German, some people say man in English. So different language, people get confused. Are we talking about the same?
Manushya.
হ্যাঁ বলো বলো কি বলছো? Okay, so some people
স্যার
say,
কি বলেন জায়গাটা আরেকটা বলুন।
so some people
সেটা শুনতে
say,
পাইনি একটু আগে।
so in different languages man is called differently. In Bengali you call them as manush, in maybe Urdu you may call it as insan. So like all these things there are different. So how do you know that manush and insan are the same thing? You need to have a common language, and that common language is you give a scientific name, Homo sapiens. So systematic biology is the field that provides scientific name for organisms. It describes them. So for Homo sapiens you have got some characteristics: 2 hands, 2 feet, you are standing on 2 feet, you have got a big brain, which is taking lot of informations from my class. So all these things, there's specific characteristics. You are writing about Homo sapiens. Preserves collections of them. In museums you'll find mummies of human beings. Okay, so these are collections preserved. For example, you go to any laboratory, you'll find specimens of animals, earthworms and different animals. They are preserved, where you have to do identification. So this is also a part of systematics, preserve collections of them. And they provide classifications. But next is they provide classifications for the organisms. That is, they classify organisms into different class, order, family, all these things. They give this classification, which kingdom it is, which sub-kingdom, which phylum, which class, order, all these things. This is done by systematics. The next thing which systematics does is it investigates their evolutionary history. That is, how close. So you have classified, you have given name, you have a cat and a dog, you have given them a name, Felis domestica, and you have given them Canis familiaris for the dog. You have given them 2 names. You have given some characters of dogs, you have given some characters of cats. Now, what is the relationship, evolutionary relationship of cats and dogs? Do they come, how are they related with each other evolutionarily? Originally, what was that? What evolved into cat and what evolved into dog? So you find this relationship. This also is done by systematics. You make a cladistics or cladogram. You draw an evolutionary diagram with lines you can show. I will explain this today itself. What you also do is you study their environment, whether they stay on the trees or whether they stay on the water, whether they stay on the land. So you're talking about their environmental adaptation also. So taxonomy is only that part of the systematics. It considers point A, point B, point C and point D. In taxonomy you don't study about the evolutionary histories and you don't study about their environmental adaptations. So this is the difference between systematics and taxonomy. Taxonomy is a part of systematics, but systematics is not a part of taxonomy. You can fit taxonomy inside systematics. Okay? Taxonomy is part of systematics, but systematics is a more broad term. In between there is taxonomy. In taxonomy you don't study point number E and point number F. So in systematics it's the science of biological diversity. It reflects phylogeny, that is the evolutionary history, and it is based on cladistic analysis. We will define it shortly. And you of course have the taxonomy. You identify, name and classify. Carl Linnaeus gave the binomial nomenclature. You have the genus and the species name, Homo sapiens, Panthera tigris, Felis domestica. Felis is the genus name, species name is domestica for cats. Okay. Then you have something known as taxonomic rank. Organisms are classified primarily according to a system of 7 ranks. They are species, genus, family, order, class, phylum, kingdom. You can you start with kingdom. Kingdom, animalia, animals, organisms are able to move on their own. Chordates, animal with a backbone. Mammals, chordates, we are calling them as mammals because they have hair and young have milk, drink milk. And you have primates, mammals with collarbones and grasping fingers. Mammals, hominids, primates with relatively flat faces and 3-dimensional vision. They are hominids. You have the species Homo, that is hominids with upright posture and large brain, that's a genus. And sapiens with high forehead and thin skull bones. So therefore Homo sapiens, human beings. So this is how you can classify a human being. The last day I explained it about a dog, now I have classified a human being. So each level, this is known as a taxon. A species is a taxon, a genus is a taxon, family is a taxon, order is a taxon, a class is a taxon, a phylum is a taxon, a kingdom is a taxon. So you have each of them is a taxon. And a hierarchy is kingdom is on the top, then followed by phylum, then followed by class, then order, family. This is known as hierarchy. Jaise on the top there is a principal, below the principal there is a vice principal, below the vice principal there is a dean, below the dean is the head of the department, below the head of the department are the teachers, below the teachers are the students. It's like this. This is known as a hierarchy. In a same, you find a hierarchy. So this is a taxonomic hierarchy. On the top kingdom, under the kingdom is phylum, under that is class, under that is order, under that is family, under that is genus, and under that is species. So to remember the taxonomic rank, this 7 ranks, King Philip cried out for goodness sake. K is for kingdom, P is for phylum, C is for class, O for order, F for family, G for genus, S for species. So King Philip cried out for goodness sake, you can remember that. So this is that of a dog, kingdom Animalia, phylum Chordata, class Mammalia, order Carnivora, family Canidae, genus Canis, species Canis familiaris.
But sir, what's about subphylum, subkingdom?
That is further classification. You can classify them further. But the basic 7 ranks are all these 7 ranks, the basic ranks. That is further you can classify, but that is in between. So in a class you can divide, this is the headmaster, these class are the general candidates, these are the reserved candidates, these are the OBC candidates. You can divide a class also like this. Similarly, you can do that further wise. So that is, but the basic 7 ranks are these 7 ranks. So the King Philip cried out loud. Okay, all the 7 ranks. So the next we come across to the taxonomic characters. So what are taxonomic characters? Taxonomic characters are the taxonomic attributes. Attributes are हिंदी में जिसको कहते हैं मैंने बंगाली ते जाके बोले वैशिष्ट्य, स्पेशलिटी. ओके, so
characteristics.
Yes, exactly. We are talking about characteristics only, and so বৈশিষ্ট্যটা যেটা কথা হচ্ছে বৈশিষ্ট্য or speciality you can say. So taxonomic characters are the taxonomic attributes that can be used to provide evidence from which relationships, the phylogeny between taxa are inferred. So basically, why are you classifying? So on the basis of what are you classifying them? Basically, all what I said. On what किस बेसिस पे इसको डिवाइड कर रहे हो अलग अलग গ্রুপ में কিসের ভিত্তিতে তুমি এটাকে ভাগ করছো. So why are you dividing it on what basis, on what characteristics, or on what attributes, or one speciality? So these specialities, they are under this person is a very famous person. His name is Ernst Mayr. So Ernst Mayr essentially it was a famous evolutionary biologist. So he gave certain characteristics. He divided it into morphological characteristics on the basis of morphology. Morphology is external. I will talk about it. Then he gave it into physiological characteristics. Then you have ecological characteristics, ethological. Ethological is behavior. Ethological characteristics mean behavioral characteristics, and geographical characteristics. So morphology means your body shape. I will talk about it. And then the fifth characteristic has come, and that is known as the molecular biology or biochemical characteristics. This is a cartoon. Okay, morphological characters. So what are morphological characters? Here, size, shape, color, etc. are morphological characters of an organism or its specific parts. Morphology describes structural features. So, animals whose body is divided into head, thorax and abdomen. Head, this is an insect here, the head, this blue region, this blue part, this is the thorax, and this region is the abdomen. And those who have 3 pairs of thoracic legs. So 3 pairs of thoracic legs are present, and they are grouped into insects, class Insecta. So this is a this is a morphological character. Then you have physiological characters. Physiological characters can be classified as all vertebrates where respiration and breathing takes place through gills. Like in fishes, it's taking place through gills. They are aquatic vertebrates. While the vertebrates where respiration takes place through lungs, they are terrestrial vertebrates. So terrestrial means living on the land. So they are living on the land, they are terrestrial vertebrates. And they are living in the water, therefore they are known as aquatic vertebrates. So you are classifying immediately, they are grouping them differently. Then you have the ecological characteristic. These are the polar bears. They live in the poles on ice and very cold climate. Their habitat is different. And then you have the grizzly mountain bear, brown bear, which live on mountains and in forests of the mountains. So they are the brown bears. But so there you can divide these 2 bears. Then you have the ethological characters. Ethological characters. So what are ethological characters? Ethological characters, just 1 second. There is a problem which has come over here. It has got copied over here. Okay, let me go back to the slideshow. Okay, ethological characters means this is Drosophila melanogaster. This is one species of the fruit fly, and this is the picture of Drosophila simulans. So both of them, they look same, but they are different. They have got their different species name. They don't mate with each other. Both are males, but they don't mate with each other. One reason is they have got a courtship behavior. They have a particular, the male has got a particular dance by which it impresses the female, so that the female will mate with the male. So simulans have got a different dance. Maybe this follows a rock and this follows Bharatnatyam. So both of them have got different dance pattern. So if a Drosophila female, melanogaster female gets a Drosophila simulans male dancing, the female will not understand that the male is wanting to mate with her. So it will not respond. The same thing happens. Only a Drosophila male, when it dances in a particular pattern, the female will understand, okay, this male wants to mate with me. Then the female will mate with the male. But if it's a simulans male, it will not. So by this you can classify there are different species. Drosophila melanogaster is a separate group. Drosophila simulans is a separate group. But they have got very lot of similarities. Then you have the geographical characteristics. For example, in Asia you have a type of elephant living, and then in Africa you have a different type of elephant living. Both of them are elephants, but they have got certain small differences, like the ear. It's a huge ear. African elephants, the Indian elephants or the Asian elephants, they have got small ears. So, but they are in different geographical region. One is in Asia, one is in Africa. So, based on the Asia and Africa, they are divided into African elephant or Asian elephant. Then you have molecular biology and biochemical characteristics which are used. So, we know that in our cell inside the nucleus we have DNA. DNA is deoxyribonucleic acid, and we also know that DNA are the carrier of information. That DNA, whatever is present in the DNA, it describes what type of organism or animal which we are. So if you compare a particular DNA, like known as ribosomal DNA, which forms normally ribosomal RNA, this particular DNA, if you compare the sequence, all DNA is made up of adenine, thymine, A, T, G, C. There are 4 nucleotides which are present. So if you compare them, so you can find that the sequences, on the basis of similarity and dissimilarity using the sequence, you can find that they can be grouped into bacteria, archaea and eukarya. So you can use molecular characteristics of ribosomal DNA of different bacteria, archaea, eukarya, and then you see similarity and dissimilarity. You can find that the bacteria are present in one group, the archaea in red in another group, and eukarya in another group. So you can use molecular biology and you can classify these different groups. Now we are talking about a new
Sir,
thing.
what, sir, what is the main difference between ecological characters and geographical characters?
Geographical character are basically different geographical region, okay, and ecological is different habitat. One lives in ice and one lives in the mountain. একজন বনে থাকে, একজন বরফে থাকে. They are also geographically separated, okay. বুঝতে পারলে? Polar bear can also, polar bear is also found in the it is found in the poles, and bears are found. You can also give their example as geographical difference. Fine, but they are also but their habitat is also different. Okay, polar bear, but basically they are overlapping. You can say you are overlapping, but you have classified polar bears and the mountain bears on the basis of their habitat. One lives in the mountain, but both of them are eating the same thing. Fish, দুজনেই মাছ খাচ্ছে. But both one is in the mountain, one is in the lives on ice. It is also geographically separated. One is living in Siberia and the poles, one is living in North American forests. So they are also ecologically different and they are also geographically different. Same with elephants. But if you this is even more prominent, you can find among the tigers. The Russian tiger is different from the Indian tiger. The Indian lion is different from the African lion. So they are geographically they are separated, as well as they are, but for lions, the Indian lions who are present in Gujarat in the Gir forests, and the lions in the African savanna grasslands, basically their habitat is the same. So they are not ecologically, they are differentiated. They are differentiated from the basis of their geography, because both lives in the Gujarat grass. One lives in the Gujarat grasslands, while another lives in the savanna grasslands of Africa. So in this case, you cannot say that they are ecologically different. They are only geographically different. One lives in Africa, one is in India. So let us talk about alpha taxonomy. What is alpha taxonomy? Alpha taxonomy, it is the discipline of detecting. That is, detecting means to identify. Pehchanna. Detecting, describing. You give a description, you write down the how do you describe one thing, একটা characteristics or বৈশিষ্ট্যগুলো লিখলে, you are describing it, and classifying new species. Then you are classifying it into kingdom, phylum, class, order, genus. Then you are classifying it. This is done by alpha taxonomy. So you see a species. This is the very initial. First you see a new animal, say you see something like a dog. First thing, you identify the dog, that is detect the dog. Then you describe the dog. Then you describe what are the characteristics of the dog, and then you give a name to the dog. So which involves identification, naming and characterization. So first identify, give a name, and then write some characters, that is describing. And so you have finished your alpha taxonomy. Alpha taxonomy does only this part. Next comes beta taxonomy, that is the second stage. So you in Asansol, you find a dog. Somebody else in Calcutta finds that another something similar to a dog, a fox, say for example. So now what you do, beta taxonomy, what they do is they try to find the relationship between the dog and the fox, how they are similar and how they are different. So you have one alpha group, alpha taxonomy group 1, that is dog in Asansol, and second alpha taxonomy group, that is a fox in Calcutta. So you are trying to find out the relationship between dog and fox. So this is what it relates the search for. So you place both of them under order carnivora. You see that both of them eat meat, so they are carnivora. So this is how you use beta taxonomy. After beta taxonomy comes gamma. So you classify, for example, some few insect order like your Drosophila. In Drosophila you find there are lots of Drosophila species. That is Drosophila simulans, Drosophila melanogaster, Drosophila willistoni, Drosophila ananesi, Drosophila blah blah. There are thousands of Drosophila species. So you try to find out an evolutionary relationship between Drosophila species. Which species, how close is Drosophila simulans with that of Drosophila melanogaster? Or how close is Drosophila ananesi with that of Drosophila melanogaster? So it has been found that Drosophila melanogaster and Drosophila simulans, they are very close with each other, and Drosophila ananesi is far away from each other, from Drosophila melanogaster. So that is this is what is studied in gamma taxonomy. Drosophila ananesi is the fruit fly which you find in India on top of your banana. If you are eating a banana right now, the fly which you see on the top, that is most probably that is a Drosophila ananesi which is flying around in India. Melanogaster, very rarely you will find them, but you can find some melanogaster, but normally I have seen melanogaster in laboratories only. What you see on top, the fruit fly which you see with two red eyes, that is Drosophila ananesi. But ananesi is far away from, by relationship, if you compare the DNA, it is much more far away. Even their characteristics are different. The morphology of the genital organ of ananesi is not similar to that of melanogaster, so they cannot mate with each other. So here it is. You have alpha 1, 3 species, alpha 2, 3 species. This is alpha taxonomy. You name, describe and identify them here in alpha taxonomy. In beta you try to find the relationship among them. And after having them in one group, you try to find the relationship of the different organisms in gamma. So in alpha taxonomy, you identify the species, describe the species, giving characteristics and give a name according to binomial nomenclature. In beta taxonomy, you further group the identified, nomenclatured and described species, but this is what you did in the alpha taxonomy, into different groups or same groups. You can keep them together or different on the basis of differences and similarities in taxonomic characters. What are the taxonomic characters you have studied? Morphology, ecology, geographical, molecular, biological, all these are taxonomic characteristics. And in gamma taxonomy, the evolutionary relationship between the groups classified by alpha and beta are determined. These are the groups classified by alpha and beta. You are trying to find out the relationship between them, evolutionary. Then another taxonomy is omega taxonomy. In omega taxonomy is the modern experimental taxonomy. It was denoted by a scientist known as Epling, in which taxonomic activities have been enriched with data from ecology, phytochemistry. Phytochemistry is plant chemistry. Phytogeography, phylogeography, that is geography, biogeography you can call them. Animals living in different regions, you study animals which have got a different biochemical compositions. Cytogenetics, that is inside the cell, the chromosomes. You are looking at the chromosomes of different organisms. And physiology. coupled with adequate computation. So you also use your computers to find out the difference and similarity between DNA sequence. So this is the modern system of taxonomy, which uses all the different approaches to study. You have to know the definition of all these things. So this is how they are specified. So species C, D, E, F, G, they are all the DNA sequence. These are the DNA sequences. They are seen. You see that here there is lot of differences. So they are, you can see there species C and species D are close to each other. But the species C and species E, they are further away from each other. They have branched away much earlier. So they are more far away from each other. Then you have cytotaxonomy or karyotaxonomy. In cytotaxonomy or karyotaxonomy, you study chromosome. This is known as polytene chromosome. This type of chromosome is present in the salivary gland of your Drosophila. This polytene chromosome is present in the salivary gland of Drosophila. You have saliva means the saliva which comes from your tongue. In the saliva you have these types of chromosomes. So this is how the salivary gland chromosome or the polytene chromosome looks in Drosophila melanogaster. In Drosophila simulans it will look different. Maybe this arm will be shorter. So, oh, the arms are, this arm is longer and this arm is shorter. Oh, shorter arm, this is simulans. Oh, longer arm, this is melanogaster. So this is how you use the chromosomes to classify or organisms. So chromosome sizes are different. So one is in simulans, one is in the melanogaster. Then you have biochemical taxonomy. The greater the similarities in certain organic compounds, the more closely related the organisms are. So organic like DNA or RNA or proteins, more similar the protein sequence or more similar the DNA sequence, the closer they are. More different the DNA sequence, the more farther away they are. So humans and chimpanzees have got a very close DNA sequence relationship, so they are very close together. But humans and gorillas have got a less similarity, so humans and gorillas are different. So the closest animals to the humans are the chimpanzees. Another biochemical are the phosphogens. So this phosphogen basically are used in the physiology. They are used in muscle contraction. They give energy to the muscle during contraction. They give high energy phosphate energy. So you have creatine phosphate. This is present in higher organisms. From echinoderms to us mammals, we have creatine phosphate in their muscles. But below echinoderms, in mollusks, in insects, you have arginine phosphate. So those who have creatine phosphate, they are classified in a different group. Those who have arginine phosphate, they are classified in a different group. Now this is an overview of the different systems of classification. You have the linear system of classification, two kingdom classification, Vegetabilia, Animalia. You have Ernst Haeckel's classification, Protista, three kingdom classification, Protista, Plantae, Animal. You have Shatton's, that is prokaryotes and eukaryotes, two empires. Then you have the Copeland's classification, four kingdom, Monera, Protoctista, and Plantae and Animalia. You have then the five kingdom of Whittaker, Monera, Protista, Plantae, Fungi, Animalia. This is taken nowadays. Then you have the three domains, the bacteria, archaea, eukarya. This is done by molecular classification by Wurst et al. I've shown the picture just some time back. Bacteria, archaea and eukarya. Then you have the sixth kingdom, bacteria, protozoa, chromista, Plantae, fungi, Animalia. So there are different types of systems of classification. In my opinion, remember Whittaker's, Carl Wurst and Linnaeus. Remember these three, that is good enough. The others are not required. There are also one place where even they tried to put virus also together. But you, as I mentioned before, you cannot put viruses together. তারপরে কে কেউ কিছু বলবে?
Sir, whose classification you have to remember to us?
বললাম তো Wurst's five kingdom, three domain and two kingdom. Only these three classification you have to remember. So the next thing is
Sir, what is
Ah.
the chromista?
they are
Chromista, what is
chromist means they use colored pigments as the respiratory or pigments. Instead of chlorophyll, they use different colors. You don't have to remember chromista example, but chromista means chrome means color. So this group of this protozoans, they have got colors. They release light. They have placed under foraminiferans right now in our five kingdoms. They have different colors. Okay. So now let us come to the last part of today's talk, that is cladograms. Cladograms, they are diagrams that show the evolutionary relationship among a group of organisms. So you see the sharks here, you see rayed fin, this is the chondrichthyes or the ostrichthyes, the ray fin fishes. Then you have the amphibians, you have the primates, the rodents, the crocodiles, dinosaurs, birds. So how do you classify organisms and how do you show the evolutionary relationship? You have invertebrates. Whether you have if you have a vertebrate, then you are vertebrate. So all of these organisms, they have a vertebrate. So the first character they are using is the vertebrate. So you branch it out as vertebrate. Then you discover whether they have a bony skeleton. So these chondrichthyes, they have a cartilaginous skeletons, these sharks. So they are separate. So you make a separate line for them. Where those who have bony skeletons, they are the people with bony, the individual or the organisms with, not people, organisms with bony skeletons. Then again, whether they have 4 limbs or not. So fishes, this ray fish, they don't have 4 limbs, so they are separated out. So these are the organisms with 4 limbs. Then you see amniotic egg. That is special egg has got a structure. Chorion, amnion, allantois, they are a special structure. So they have got an amnion inside their eggs. So the amnions, who are amnions? So what separates out over here? Amnions, the amphibians don't have amnions, so they are not amniotic. So the reptiles are amniotic like crocodiles. Then you go for hair. Who have hair? These primates and rodents and rabbits have hair, so they are separate group. And then you have 2 post-orbital fenestrae. In their skull they have got 2 holes. And that is why the crocodiles and birds and dinosaurs, they are placed in one group. Again they have feathers, so you can again separate the birds. So this is how a cladogram is made on the basis of certain characters. First, what you have, you are given these organisms. I give you 5 names, 1 2 3 6 7 names. One is shark, one is ray fin fish, amphibians, primates, rodents and rabbits, crocodiles, dinosaurs and birds. And then I tell you, okay, make a cladogram. So first, what you consider, okay, all of them, they have vertebrates, so they are vertebrata, so they are together. Then you see who have got bony skeleton. You see that only these have got bony skeleton, so you place them together in one group and this as another group. Then after that you see, you check who have got 4 limbs. You see only these have got 4 limbs. Even birds have got 2 wings and 2 legs, so 4 limbs. Next is who have got amniotic eggs. Oh, only these have got amniotic eggs. So you make this into another group. So like this you make a cladogram on the basis of taxonomic characters. So with this I sort of end my class today. Okay, and hope you were able to understand this. I will tell when my next class is on Wednesday. Most possibly on Wednesday I will try to do it in the morning. But since we are busy making the new first year your accounts in online teaching in the college. So for that I'm a bit busy, so I cannot exactly tell the time. But very soon that will be finalized. So on Wednesday I will take the next class. Okay. So do people have any problems? Please do ask if you have problems.
No, sir. No, sir.
Nisab, Al-Shifa, Afshar, batao if you have problems. Shoham, if you have any problems.
No, sir.
No problems. Okay. Then I will send the recording of this this class very immediately after I finish the recording. Okay, then I finish the class. Bye bye. Have a nice evening then.
Okay, sir.
Okay, bye bye.